So I want to show you now what is sort of our state-of-the-art thinking about what is actually needed to build the machinery at a gene to allow it to be expressed and transcribed. And the term I want to introduce you to is the pre-initiation complex. And it's pretty much what it says. It's the complex of multiple subunits that has to essentially land on the promoter of a gene, which will be designated for later expression. And this is a process that is probably quite orderly. That is, there's an order of events that happens, which we, by the way, are not entirely sure exactly what the order is, or even if the order is the same from one gene to the next. But we can kind of see where it starts and where it ends up. And the pathway in between, I would say, is still a little bit murky. And the story here, again, starts with a little snippet of DNA called the Tata box, which I already introduced you to briefly. It's an AT-rich sequence which sits at the 5' end, or the beginning of many genes, but not all genes. Maybe 20% of the genes might contain this AT-rich region. And that AT sequence is the signal, or a landmark, if you like, for a particular protein to bind to it. And that protein is called, not surprisingly, the Tata binding protein, because it's the Tata sequence. And so this represents a second class of transcription factors. These are not the type that I just introduced you to, which are going to be different for every gene. The Tata sequence is present in a very large number of genes, so it can't be gene-specific. But it turns out to be very crucial for our understanding of how gene regulation works. So you start with a Tata binding protein finding a Tata box. We later found out that the Tata binding protein rarely functions on its own and has a bunch of friends that we call TAFs, for TBP-associated factors. And now you're talking about an assembly, a multi-subunit complex of almost 1 million Daltons. There are somewhere between 12 to 15 subunits in addition to the Tata binding protein that make up this little complex of proteins that kind of travels around together. And this is found in most cell types. And later on, I'll show you in a subsequent lecture that not every cell type might have exactly the same complement of these subunits, but many of them have this prototypic complex. Is this enough for building the pre-initiation complex? Unfortunately not. It turns out that there are a host of other, I'll call them ancillary factors, in addition to the multi-subunit RNA polymerase itself, that are necessary for you to build up an ensemble that is necessary to form an active, ready to activate transcriptional pre-initiation complex, or the PIC. And this is kind of the picture we're getting to, and even this picture with many, many colors and many, many different polypeptides, you know, that adds up to probably greater than 85 individual proteins that all have to kind of fit together like a jigsaw puzzle. It's probably not even the whole story. You'll notice I still have one big red question mark there, because I think as we begin to study specific cell types and specific processes like embryonic development or germ layer formation, additional components that are not present here in this prototypic pre-initiation complex will come into play, and that's a subject of a subsequent lecture. But already you can tell that the transcriptional machinery is anything but simple. So can we get a better idea of what transcription might actually look like in what's happening when a transcription process takes place? So let me first of all say that I'm going to finish my lecture now with a little cartoon, which is our attempt to imagine the events that take place when you form a pre-initiation complex, you bring regulatory proteins to the activated gene, and what happens during this process. Now, keep in mind that this is at this point mostly a cartoon that is in our imagination, and only parts, or if any, of this is probably real. But it gives you a sense of the complexity of the transactions that have to take place just for one gene to transcribe and express itself. So let me show you the movie, and then we'll finish just by keeping in mind that There's much to be learned, and in my next lecture we'll go into the selectivity of this process in specialized cell types. So now let's see what this sort of this cartoon of transcription looks like. So we start off with DNA with some pre-assembled TF2D molecule, and along comes this other green molecule, which is actually a cofactor, which then forms this very large complex with RNA polymerase. And then a distal activator protein came in and activated the process. And this molecule, this bluish molecule that's moved away from the complex is actually the RNA polymerase, and that little yellow sort of bead on a string is actually the RNA product. So that gives you a sense of, things have to happen quickly, and yet it involves many, many molecules having to assemble and then disassemble to give you this reaction to happen. And in my next lecture, we'll go into more specific aspects of this reaction, and particularly during embryonic development and tissue-specific gene expression.